Hello, my name is Bradley Wilkerson. I'll be working on question 2 of the group project practice problems. Um, so for A, we have TF2D as what binds to the TATA box, while the sigma factors bind directly to the 10 and 35 sequences. And then for B, we have this long name, the guanylitran site phosphorus. Um, and this is in the eukaryotic enzymes that links to the 5 carbon and 1 nucleotide to the 5 carbon and another nucleotide. And then for C, we have group 1 introns and group 2 introns. Group 1 introns use the nucleotide guanine in the cofactor during catalyze, while group 2 uses adenine as the cofactor during catalyze. And then for D, we have CPSF, which is the endonuclease that assists in the terminating transcription in eukaryotes. And then for E, in lesson 9, we talked about the joint transcription factors. And then TF2B is a GTF which acts as a bridge, the subunit to link TF2D to TF2F and the RNA primase 2. And then TF2H is another GTF which acts as a acts to phosphorylate the active RNA polymerase 2. And then for F, we have SRNPS and small nuclear RNAS. And those are just, the first one is a subunit of spliceosome. And then the second one is part of the spliceosome, which is the proteins and the small nuclear RNA.
I'm Hadley Pinder, and I'll be doing problem 3. And so it says this question concerns the proteins involved in transcription and RNA modification. Part A, indicate 2 proteins in either prokaryotes or eukaryotes that act as a helicase to separate 2 DNA strands during transcription. And so I got TF2E because that's what causes it to be an open complex, and TF2H. And then for B, says indicate 1 protein in prokaryotes that acts as a helicase to separate RNA from the DNA during transcription, and that's rho protein. Then C says indicate 3 molecules in eukaryotes that function during RNA modification as ribozymes. We got guanosine nucleoside, adenine nucleotide, and spliceosomes. The part is the subunit SNRNP. And then indicate 1 protein in prokaryotes that acts as a transcription factor. I'll just do 5 too.
Okay.
Maybe we can turn around.
Yeah.
That acts as a transcription factor, and this would be the sigma factor. And for the last one, says indicate 1 enzyme in eukaryotes that synthesizes a long string of identical RNA nucleotides, but does not use a template DNA strand for RNA synthesis. So the poly A polymerase adds a bunch of adenines on as a tail. Okay, for 5, says, gives us the list of proteins and nucleic acid sequences. And it says which of the above are found in E. coli, and it would be the RUT, -35, and the NUSA. And then for part B, which of the above is in yeast, and it's -25. The enhancer, silencer, group 1 intron, GC box, TF2D, and CNRNP. And then C, which is above, is involved in transcription initiation. We said the enhancer, silencer, -25, -35, the GC box, and the TF2D. Which of the above is involved in transcription termination? RUT and NUS-A. And which of the above is a DNA sequence? So silencer, enhancer, -25, -35, GC box. And as far as the RUT one goes, it was in the picture on the PowerPoint to be a part of DNA, but it wasn't in our notes, so we were tentative. Don't just turn around or stop it.
I'll just come up here and just
Okay.
do it. I'll just redraw and talk
Okay.
about it as I go. Okay, I'm Donovan Wixom, and I'll be doing question 4, which I'm going to pull up really quick like. So it says, you hypothesize that a eukaryotic gene called your favorite gene contains 4 introns, or 4 bits of DNA, or the RNA, 4 bits of RNA are going to be cut out of it. And we're just going to do loop models for them. So part A says, the pre-mRNA for your favorite gene is hybridized to the genomic DNA. So I wish we had different color markers, but we don't, so bear with me. I'm going to draw this one up here. We have our pre-mRNA, so the introns are still in there. I'll show the introns by doing the squigglies, just because I don't have any other way of showing it. So there's 4 of them, and we'll make this the 5' end and the 3' end. So the genomic DNA has 2 parts. We'll do the template strand first. This is going to be the 5' end of the template strand and the 3' end. The template strand is going to bind with the mRNA all along here. So they're just going to look like a normal bit of nucleic acid. The coding strand is going to start with its 5' end over here, and it's going to bind with the DNA like it normally would. But when it gets to the pre-mRNA, it has nothing to bind to. So what it does is it forms an R loop, or it just kind of floats up here, isn't bound to anything, and then as soon as the mRNA is done, it'll come back down and bind with the DNA again, ending at its 3' end. So this is what A is going to look like. Again, we have the coding strand, the template strand, and the mRNA with all 4 introns. So part B says that a mature mRNA is hybridized. Well, mature means that it has all the introns cut out of it. So when we draw our mRNA, which I'll do down here, it's going to be a little bit shorter. It doesn't have the introns. So we'll just draw it about like that long. Again, we'll do a 5' end and a 3' end. The template strand is going to come along with its 3' end, and it's going to start binding until it gets to a point where the intron used to be. Now there's all this extra DNA that has nothing to bind to. So it's just going to loop up and come back down until it can start binding again. And since there were 4 introns, it's going to do this 4 times. We're going to get 4 of these loops that I'm going to have to make our mRNA a bit bigger. So we've got 4 loops. It's binding to the mRNA. That's a 3 prime. And it's going to end at its 5' end. It's going to go a little past it. So then the coding strand is going to do the same thing it did up here. It's going to start 5' end, and it's going to bind with the DNA until it can't. It's going to form an R loop. But when it gets to this loop of DNA, it has sequence that combined to it, it has the intron sequence as well. So it's going to loop over with it and bind until it gets to the mRNA, and then it's going to do another R loop. And I made this kind of small, so it's going to have to just kind of do this. And then it's going to R loop here at the end until it combined, then it'll start binding again and end at its 3 end. So template strand, coding strand, mRNA. These big loops are the intron loops, and the R loops are going to be wherever the coding strand can't bind, which there's going to be 5 spots. 4 spots. 1, 2, 3, 4, 5. 5 spots. I can count. And 4 loops. So for part C and D, basically, for part C, the mature mRNA for your favorite gene is hybridized to the genomic copy. In this case, however, the 5' most intron, so 5' most, this intron, it has a defective donor site and branch site. So it's not going to be able to be cut out. So when we come down here, I'm just going to try to, I'll just draw a new picture instead of trying to edit it. So when we have our RNA, I didn't mark B, C. So when we have our mRNA, it's going to come and it's going to have its 5' most intron, but the rest of them got cut out. So it's only got the 1 intron. So our template strand comes, it's going to start binding, and it's going to bind all the way up past this intron, and then it's going to loop for the other 3 introns. So we'll get 3 loops. Oop. It starts to look sloppy when I get over here, sorry. 3', 5', 3'. And then, like we've been talking about, coding strand's going to do the same thing. It's going to bind, R loop, intron loop, R loop, intron loop, R loop, intron loop, R loop, bind. That's a big R loop. R loop, bind. And again, it'll be bound all along these intron loops. So we'll only have the 3 intron loops, the 3 later intron loops for part C. And I didn't leave much room for part D, so we're going to erase part B. I want to leave A up to show one more thing. So this question is very similar to question C, but instead of the first one not working, it's the second and fourth one. So these 2 introns will still be cut out. So when we draw our RNA molecule, it'll come along, first one's cut out, second one isn't. Third one, I'm reading that right, making sure I'm not lying real quick. The second and fourth, yeah. So third one's cut out, fourth one isn't. 5', 3'. So our template's going to come along, it's going to bind, then it's going to loop for where the first intron was, bind for the second, loop for the third, bind to the fourth. And again, our coding strand, this is the 3', 5'. Coding strand's going to do bind, R loop, intron loop where it's binding, R loop where it isn't, intron where it is, R loop where it isn't, and then bind again. And end at 3', 5'. So that is 4. That's the last one, so I'm going to stop it.